Hi everyone, my name is Kang Jin. I'm from Cincinnati Children's Hospital. Today I'm going to present our recent work, CellDrift, which is a tool to infer perturbation responses in temporally sampled single-cell data. And this is our bioRxiv preprint. The motivation of CellDrift is that nowadays, we are more and more commonly see very complicated single-cell perturbational dataset, which usually involves in multiple time points. For example, dataset during the disease progression, organ development, or drug responses. However, there's very few methods that can be used to resolve this issue. So that's why we developed CellDrift. And CellDrift consists of two parts. The first part is a generalized linear model, which is used to decompose the perturbation responses at a single time point. And then we combine the result from generalized linear model of multiple time points and input it in functional data analysis, which is used to analyze temporal data. And with it, we can identify temporal clustering patterns or reduce the temporal complexity using functional PCA, or prioritize temporally associated genes using functional ANOVA test. And this is an overview of our generalized linear model, where we try to decompose the complexity of count data of single cell using a series of different factors, including cell type coefficient, perturbation coefficient, and their two-way interaction. With the result of the generalized linear model, we construct a series of contrast coefficients called delta beta GCP, which represent the perturbation responses for gene G in cell type C and perturbation P at time point T. And by combining a series of contrast coefficients, we can construct temporal data either in this format or in this format to represent different needs of temporal analysis. More details can be achieved in the manuscript. With the established method, we are looking for the evaluation of performance of CellDrift, and we benchmark CellDrift in two parts. One is the benchmark of generalized linear model, and the other is the benchmark of functional data analysis. To mimic real data, we generated simulated data using splatter with different degrees of perturbation responses and batch effects. And we benchmark CellDrift with other commonly used differential expression method, and we find that our method has the best performance in identifying the perturbed genes using the evaluation matrix called TPR in different parameter settings. Although the performance of FDR is not always the best in CellDrift, but the range of FDR is in a reasonable range. And furthermore, we benchmark CellDrift with other methods to evaluate the performance of identification of temporal patterns. So we simulate single-cell data with perturbation effects across time point, and we simulate different parameter settings. And our result shows that our strategy of CellDrift, which consists of the result from generalized linear model and the clustering method called FastCC means, always achieve the most robust and very good performance in identifying temporal patterns or in imputation of missing time point. So with the benchmark result showing that CellDrift achieve better result than other alternative approaches, we further extend CellDrift into real examples, including this COVID-19 example here. So this is a pretty complicated COVID-19 example with multiple conditions of disease, and multiple cell types, as well as different days since disease onset, which is a time point covariate. And we applied CellDrift in this dataset and identified 20 distinct temporal patterns or clusterings, and each of which represent a unique temporal pattern. For example, here we show 3 representative clusters, including cluster 11, which is downregulated across time points in the disease, and another cluster, cluster 17, which represent upregulated temporal perturbation responses. And their enrichment result shows very distinct pathway enrichment, which was validated by literature. Furthermore, we try to prioritize disease-associated temporal genes using functional ANOVA test. So here are 5 prioritized genes that prioritized by functional ANOVA test between the mild COVID-19 disease and the severe infection disease. So these 5 genes are more upregulated across time point in severe conditions consistently. And we applied functional ANOVA test again to another very large-scale COVID-19 example, and we found the same trend for those 5 prioritized genes that we found in the first dataset. As a summary, we successfully applied the generalized linear model as well as functional data analysis to build a method that can help us decompose the temporal patterns of perturbation responses in single-cell data, and we validate the performance of CellDrift using benchmark dataset as well as real dataset to evaluate its performance. And that's basically what I have, and thanks for listening. Welcome to my poster section.